Hi everyone. So today we are going to have a session on hands-on workshop, and we will see how we can analyze the single cell transcriptomics data in COVID-19 brain endothelial dysfunction, and also how it plays role in glioblastoma multiforme implications. So, one of the pertinent question is this: does COVID-19 associated brain endothelial dysfunction share molecular features with glioblastoma progression? So, this is one of the question which we will try to answer throughout the session through the single cell data analysis, and we will also see some of the tools which are required and how we can generate the figures and visualize the data. So we will use a paper as our narrative, and so that will act as a spine, but the goal is a practical skill, like how we can analyze a particular data set from gene expression omnibus. So by the end, you should have a reproducible workflow and a clear interpretation. So why this workshop is important? So it's the single cell data, for example, as we talked earlier yesterday. This is it is everywhere, but the challenge is turning it into a decision, like how what your data is telling, and what this single cell data is contributing to, which cell type change, which pathway shift, and how confident we should be. This paper is a good case study because it connects the clinical context, like we are talking about COVID-19, which was one of the recent pandemic we faced about, and the neurological effects and the blood-brain barrier integrity with a cancer context, like that is glioma biology. So today we will learn the mechanics, but also the mindset is like, don't just make a UMAP, ask what the comparison is being made and what the confounders exist and what would count as an evidence for a link. So our major learning outcomes will be like we will see how our data can be utilized to produce the like meaningful data and visualization, and how we can do the scientific interpretation. For example, here we are talking about how the COVID-19 and the glioblastoma are associated, and how the COVID-19 is causing this enhancement in the glioblastoma multiforme progression, which is a very rare brain cancer. So of course there are some technical skills which which we talked about yesterday, and since this workshop is meant more for those who don't have coding skills, so I have tried to make it as easy as possible, like without talking too much about the codes, so that how you can use some of the very important single cell data portal, and because those are already preset and you can easily analyze, and you can generate the data, and you can talk about your research, and you can generate the hypothesis, and you can see how it goes from there. Well, the agenda is we'll have the roadmap, that is first 50 to 20 minutes, like our workflow foundation, like loading and all this. We'll talk about the context, and also we'll not talk too much about the pseudo basic, because that is more related with the coding thing. Python, you need to know the Python coding, so we are not going too much detail about it, because I have some more alternative strategies for you to understand, which you can easily employ to generate the figures, and finally leading to a successful paper. Because this, as we should not deviate from our original talk, that how to publish a paper without lab experiment, so of course without too much hassle. So we'll talk about why we are focusing on endothelial cells dysfunction, and in the context of COVID and glioblastoma overlap, and finally we'll wrap up with a question answer session. And throughout this whole session I will keep on showing you how to generate the data. So the paper in like, if we see this paper which we are discussing here, in one slide we can talk about, like like one sentence summary for this paper is single cell analysis suggests COVID brain endothelial cells that shows inflammatory and permeability related transcriptomic changes. So you have to understand inflammation. We are talking about inflammation and we are talking about permeability. So inflammation is very important phenotype because in most of the pathological conditions the inflammation happens, and also there are some cell types which contribute to this inflammation, or even rather you can say during the inflammation there are certain cell types which change their state. For example, microglia or macrophage polarization is one of the important parameters which people generally consider. And also they talk about permeability of the blood-brain barrier, which is one of the crucial barriers, semi-permeable barrier, which demarcates our brain from other body parts. So it basically makes our brain immune free, and also it immune to other hazardous chemicals or even other things. So in this paper we are talking about like the single cell analysis and how it suggests the COVID brain endothelial cells. They show the inflammatory and permeability related transcriptomic changes. So these transcriptomic changes are generally we are talking about at the RNA level, mRNA level, messenger RNA, and some of these signals overlap with the program relevant to glioblastoma, raising the hypothesis that endothelial dysfunction could be a connecting axis. So we have to keep in mind this is primarily a computational overlap claim, not direct causal proof. We will treat it as a hypothesis generator and we will practice how to reproduce the logic. So there are some key terminologies which we have to be mindful about. So as we go through, we will encounter various terminologies. So these are some quick vocabulary which we should be familiar about. For example, cells or nuclei. So many brain data sets are like single nuclei RNA-seq, or interpretation differs slightly from single cell RNA-seq. So in that case we mainly refer to single nuclei, means each nucleus we are talking about, and single cells, overall the whole cell we are talking about, like one single cell, and each cell has one nucleus. So when we are talking about snRNA, that is single nuclear RNA, and when we are talking about single cell RNA, so we are mainly talking about single cell. And counts versus normalized expression, if there is a count of gene expression and whether it has been normalized with any particular housekeeping genes, so that counts for the normalized expression. And then there are many QC matrices like genes per cell, UMIs, mitochondrial percent. We are not going too much detail into this because we will be using all predefined or uploaded data sets. And then there are some dimensionality reduction like PCA for structure, and even this UMAP for visualization and clustering we talked about yesterday, like graph based grouping of similar expression profiles like marker genes and all of this. And there are some biological terms which we should be familiar with. For example, here is one of these is the blood-brain barrier, that is it's an endothelial tight junction system controlling permeability. So basically this BBB contains many different cell types including the endothelial cells and the astrocytes and the pericytes. Again we'll further discuss about this. And so they control the permeability of the barrier and permeability of the blood-brain barrier, BBB. And whenever there is some pathological conditions, there are chance of permeability, increase in permeability of these barriers. So endothelial dysfunction, so it refers to the impaired vascular barrier and antithrombotic or anti-inflammatory functions. So this is one of the crucial phenotype. It occurs in most of the disease conditions. And there are some innate immunity, like early inflammatory responses happens, then interferon stimulatory genes, and there are many other complementary genes which are overexpressed as a genotype for the innate immunity. Then glioblastoma multiforme, GBM, which is an aggressive brain tumor with a strong immune and vascular components. A lot of blood vessels will be there, so it is a fourth stage brain cancer. And the transcription factors, So there are some regulators which shape the inflammatory gene programs. So these are the terminologies which we'll be mainly using throughout this workshop. So that's why it's very important to know. Then conceptually, we here the conceptual flow is about the COVID brain data. We'll use the COVID-19 brain data, okay, and we'll identify the endothelial state changes. So first of all, what we'll do is we'll take COVID related brain data. So those brain data which the patients who had died from the COVID-19, we'll take those patients' brain data, and we'll identify the endothelial state changes, how it changes, because we are claiming that there is an endothelial dysfunction. Then we'll check about inflammatory programs, okay, and we'll identify the blood-brain barrier (BBB) integrity shift. Then the GBM data will move to identify malignant cells and microenvironment populations. And the key link is, do these genes or pathways highlighted in COVID endothelial cell appear in GBM cell states or microenvironment programs? We are not proving COVID caused GBM. We are testing whether there is a shared biology that could motivate follow-up experiments. So basically we are trying to see if there is some overlap. So of course there might be some association, and that can be revalidated by using some experimental techniques like in vitro or in vivo, by using in vitro or in vivo experimental techniques. So here we'll have a glance at the data set. So we are working with two GEO data sets, gene expression omnibus associated data sets, and referenced in this paper particularly, one for GBM and one for the COVID inflammatory brain tumors. So we'll check one by one. For each data set, always ask what are the samples, how many patients, what tissue regions, and what technology differences in sample preparation and sequences depth can dominate results. So integration and batch correction matter, and also we'll note the paper's choice of comparison. For example, here we are talking about COVID versus non-viral control, GBM cellular compartments. So let's move on to that particular, we'll come back to this slide again, and let's move to that single cell portal which I have been discussing so far. So I'll just share my screen with you. If you all can see this, this is a anyway, throughout this workshop we will also be utilizing this particular website. It's a very important website, GeneCard, where we can find whatever gene. For example, if you are focusing on microglia, okay, microglia is a type of immune cell which is prevalent, which is found in human brain, which is found in brain generally. And so if we, and there is another cell type, that is macrophage, which is found in mostly peripheral blood. And this microglia and macrophage, they are also referred as brother cells among each other. So if we are trying to see the microglia, what are the most prevalent genes, highly expressed genes in this particular cell type. So here you can see like TREM2, BDNF, AS, or even CSF1R. So these are the like top genes which we have. Even see here you can see SNCA, synuclein alpha, which is also related with with dementia or even Alzheimer related. Then SOD1, these are also related with this neurodegeneration related disease. So we will directly go to this single cell portal. So this portal is very important. It's developed by Broad Institute, which is a shared institute by Massachusetts General Hospital, that's Harvard, and the MIT, Massachusetts Institute of Technology. So it is a shared institute by Harvard and MIT. And so here we can see all the data sets. See here you can find, for example, previously I have checked for glioma here. If you see here, so they feature total 969 studies, and they have like, oh my god, so they have like almost 742 lakh 3 4 5 6 7, so almost more than 7.4 crore cells we have here, more than 7.4 crore cells overall. So as we have been focusing on gliomas, we will directly search for glioma. So we can go to check them one by one. So this data is for glioma, and they are from the brain, and the species is human. And then like you have both male and female here, and what are the library preparations they have used, all they have mentioned here, And if suppose if we are trying to use this particular data set, we'll check this one, and we'll see what kind of things they have. So here you can also read the summary. For example, here the related abstract of that paper has been provided here. And of course, if we search this, we will also find out which paper is related with this particular data set. So here we found it. See, so this is published in, you can see it's published in Nature. So this is a very good paper, and it was published in 2025. I think simultaneously they have published even they have also published previously in 2023, as we saw in the previous search option. So anyway, so this data set is related with this paper, so they have generated the data set. So here is one option there for exploring the data. So you can go to explore option, and you can see what all clustering they already have. For example, they already have the UMAP cluster here, and you also have like what are like different annotations. So we can from here we can select what are the annotations they have. For example, if you see, they have like total tumor annotation. So annotation means they have already named based on the their top overexpressed genes. So we are going to select all genes here, and we will not do the subsampling. So finally we will check. For example, we have to first of all we have to check whether this data set whether the clustering is true or not. For example, let's see the myeloid. Okay, so myeloid is the green color they have put. So let's go to this gene card, and we will first check whether they have like whether their cluster is correct or not. We'll check myeloid. Okay, so I think my spelling is wrong. I think we have to go back and check. MYELOID, MYE. We can check here. So this is one of the top gene for myeloid. Okay, so let's check this particular genes in this. So we can paste here CEBPA. So let's check whether it is highly overexpressed here or not. Okay, this is true. See, it has a very high expression in this. So we can say again, if you want, you can validate some other cell type also. So this has the highest one. For example, we can go to even distribution. It will create a violin plot, cell type specific violin plot, and it will give you how much expression of that particular gene, that is CBPA, which is one of the highest overexpression gene in that particular cell type, as we checked from the gene card here, CEBPA. So we will check whether they are highly present or not. So it is still loading. So by that time we can also explore some other options. And so here also I have another data set open. So this is also from glioma. Okay, single cell analysis of human glioma. Let's check in this data set, CEBPA. So here it is CEBPA. So here we'll go to the annotation first. Otherwise, without annotation we cannot understand. So sometimes they keep the name as assignment. They have already assigned. So if this is the myeloid, so this particular cluster should have the highest C. So this shows that this clustering has been done well enough. Okay, And again here also you can check the distribution. Again it is taking time, so to save time we can go to check. So again this is also loading. Anyway, so we'll come back later here. So overall we found that this is fine, you know. If we want to verify in another cell type, we can check even let's check malignant cell. So one of the malignant means this cancer. So one of the important gene for cancer is EGFR. Let's check where does this EGFR lie. So of course all the blue regions should have the corresponding highest expression of this EGFR. Okay, so this shows, so this has the highest one. So of course, so these are the like cancer cells. And suppose if you want to verify some other one like T cell or oligo, if you want, we can go to gene card and we can check oligodendroglia. So oligodendroglia is also one of the important cells. So PLP1, so we'll go to check whether PLP1 is okay. So here this is already loaded here. So CEBPA. So if you see we can go to this assignment. Sometimes it takes little bit of more time. So anyway, we will check the PLP1 here. So PLP1, okay. So now we can see that PLP1 is okay. So it's still loading. So it's okay. So this corresponds to the oligo, and oligo has the highest expression of PLP1. Okay. So with this, it is quite evident that we have better clustering evidence here, and so this clustering is well enough. So this data is acceptable. So again, if you want to see the distribution, again this is it is taking a little bit of time. Either we can wait or we can just move to another one. So here you can see, and by that time in the meanwhile we can even explore this particular data set. So suppose if you want to check the differential expressed genes, so for example here all those, so even here you can find like all this type, grade, phase, sex, cluster, subcluster, sub-assignment, library preparation, all this they have uploaded here. So this is already fit in the pipeline, so you can explore just by clicking this options. And next we can go to check based on our hypothesis. So as we were discussing, okay, we'll go back to our PPT. Okay, so this data set is for the COVID, and this data set is for the GBM. Okay, so here you can see like how many like what's the cell number they have and what's the population they have. So we are not going to use SURET or this R programming tool here because that requires coding. So we are using some other alternative methods which will generate those data much easily. Okay, so we are taking example of the single cell portal. So we have to design the logic. So we have to see that this designing this logic means like we have to define the comparison precisely, and here COVID versus control or like inside endothelial cells, then seeing where those signals land in the GBM. So this avoids a common pitfall like comparing GBM whole brain versus GBM whole tumor, which would just reflect tissue comparison differences. Instead, we focus on a specific cell type and a specific gene modules. And if you remember one thing, the integrity of your inference depends on the integrity of your comparison. Okay, so for example, again we for example here we have made a claim that I mean not exact claim, but we are trying to analyze if there is some association or overlap between this COVID-19 and glioma, whether the COVID-19 causing a surge in certain genes which are related with the inflammation, and then it is even causing which are all associated with the glioma progression. So for that we'll again go back to that portal and we'll check one by one. So for example, we will find some data set related with COVID-19. We will find some related with the COVID-19. Okay, so here we will see there are many. This data set is from Columbia University, so we have pretty good number of cells here. So we will suppose just take this one for now, and we'll check, or even there are many others. Okay, so here we got it. So we will check the different disease ontology. So disease ontology will tell you about the COVID-19, long COVID-19, those who had, or even those who had the normal healthy people, healthy population, and then those who had some respiratory failure. So suppose we are trying to check whether the inflammation has happened or not. So for that we'll check inflammation related genes here. So for example, this NLRP3. So we'll go to this one and we'll check whether NLRP is enhanced or not. So there is a filter plot you can do. For example, you can clearly see how much is the difference between control versus other groups. So you can find there are some, even you can check how much is the change from this male and female. So since we have chose disease ontology, we can also do some distribution plots. So here you can see in the long COVID-19 they have much less this particular inflammatory marker, and this is little bit of high. For example, this is around this is around 2.4, the normal y-axis, or even, and the normal maximum is 3.753, and this one is around 3 points. So this is little bit of high, and then this is also less. So overall, and you can also find out which cell type has the highest expression. So if you see these are the blue, so if you want you can check the even assignment. For example, we have to check category. Sometimes they put the name with the category. So here you can see they have pretty much mixed population here, and so these are some mixed population here, you can see. And similar way we will try to check in our COVID-19 in our glioma. For example, what is that? NLRP3, and we'll go to this. Okay, so inflammation. So this should be NLRP3. This is high in mostly in the myeloid cell. This is myeloid cell type, and also this pink color. What does this pink color denote? Okay, T cells. So mostly they are highly expressed in the immune cells like T cells or even the myeloid cells. And again, if we want to see the distribution depending on some, for example, if we want to see based on the disease ontology or something like diagnosis, let's see based on the diagnosis how much it is showing. So with this way we can easily find out where the shift is happening. Like for example, as we talked about, there is a high inflammation of inflammation in this COVID-19 and glioma. So of course we'll check those genes are showing in a similar kind of pattern or not when this particular this disease happens. So with this way you can make a hypothesis or even generate your data visualization. For example, you can do this UMAP and TCD map, and also you can find distribution. And suppose if you have multiple genes, you can even create, for example, all this LMNB1, you can create even what you call it, dot plot. So this one will also give you dot plot option if you have multiple genes. You don't have to write any kind of code, you just have to type here, and easily very beautiful dot plot you can create here. So okay, so this is how you have the okay. So this is a little bit hanged, we have to just be with it. Okay, so now it is ready. So here you can easily have this dot plots. So with this, it shows that even depending on the diagnosis, for example, here you can see some of the genes are highly overexpressed in oligodendroglioma. So these are different types of brain cancer cells like glioblastoma, astrocytoma, or oligodendroglioma. So I hope you understood how you can make your hypothesis and how you can work with visualizing data without using even code, and you can easily generate the data, and finally it will lead to a very successful paper. You can refer to the paper which we have been discussing, and if you have any question, please direct it to me. I hope you liked it. Thank you.